Hello, my name is Adolfo Garcia-Sastre. I'm director of the Global Health and Emerging Pathogens Institute at the Icahn School of Medicine at Mount Sinai in New York. And I've been working here for almost 30 years, working on virus infections mainly, especially influenza virus. And my group, we like to call ourselves the virus fighters because we are trying to come up with new strategies to prevent viral infections, especially influenza infections. And today I'm going to be talking about the novel technologies that we are using to study and prevent influenza virus infections and what we are trying to do in this field in order to reduce the burden that influenza virus causes in humans. So my presentation is divided in three parts. First is how we use novel technologies to study virus determinants of disease. Second part is going to be how we use novel technologies to identify host therapeutic targets that can be used to reduce severe disease induced by influenza viruses. And the third part, I'm going to talk about promising, but still early on, vaccine strategies that we are using to try to come out with a better vaccine against influenza viruses, a vaccine that we like to call universal because it will be able to prevent, if it works, any type of influenza virus that is circulating or can circulate in humans in the future, as well as the ones that have been circulating in the past. So the main topic of my talk is going to be influenza viruses, and here I just saw a picture of the electron microscope that was obtained by my colleague Yoshi Kawaoka that shows basically an influenza virus in which one can see these 8 dots in the middle, which represent each one of the RNA segments of the virus. It's a segmented RNA virus with 8 RNA segments, and this is surrounded by a circle, which is composed by a lipid envelope, in which one can see some spikes, also in gray. And these spikes are the viral glycoproteins, hemagglutinin and neuraminidase, H and N, that are responsible for the names that we give to different subtypes of influenza virus, like H1N1, meaning hemagglutinin subtype 1, neuraminidase subtype 1, H3N2s, H2N2s, H5N1s, etc. Now, in terms of how many influenza viruses we find in humans, we know about influenza since 1918, with the pandemic 1918 occurred. And since then, there have been different types of influenza viruses that are antigenically different. And that means that if you get infected with one of these antigenically different types, you don't get protection against a different type. And the types that have been circulating in humans, they are, first of all, influenza B viruses and influenza A viruses. Influenza B viruses have been circulating in humans for a while. And if one gets infected with one influenza B virus, one develops good immune responses that will be able to prevent infection with exactly the same strain of influenza B virus, and the same thing happened with influenza A virus. Now, the problem with influenza is that it's able to reinfect people that has been infected before because the virus changes from year to year. And through accumulations of changes, this is what is called antigenic drift, accumulations of changes, it becomes 1 year that is so different from the virus that infected one person before that now it can evade pre-existing immunity in this individual and can reinfect. This happens both with influenza B and influenza A viruses, but influenza A virus come also in different antigenic flavors. In humans, there have been H1N1s, H2N2s, H3N2s circulating, and these are what are known as the subtypes. Each one of these subtypes contains a hemagglutinin and a neuraminidase that is antigenically very different if it's a different number. H2s are different from H1s, H3 are different from H2s. And when a new hemagglutinin comes and jumps from a new type into humans, that usually causes a pandemic. That's what happens in 1918, that H1N1s came and then caused the pandemic of 1918, continues to circulate in humans, changing from year to year, till in 1957 it came a completely different, antigenically different virus, H2N2, that caused the pandemic of 1957, and these new viruses resulted in a stopping of the circulation of the previous viruses H1N1s, not of influenza B, but the influenza A H1N1s. this virus was circulating, changing again from year to year, till 1968 that came an H3 virus, and that caused the pandemic of 1968. Again, a virus that is very different from the H2s. Each time that one of these types come to the population, what happened is that because there is almost no pre-existing immunity in humans against this new subtype, then this virus is able to infect more people than normally seasonal influenza virus, that there is more infections, and infections in general are also more severe. So there is more infections and more cases of severe infections. H3 and 2s displaced the H2 and 2s, and they were circulating till today, changing again from year to year. And in 1977, H1N1s came back to the population. This didn't cause a pandemic because they were practically identical to the ones that disappeared in the 50s. But they started to co-circulate in humans with the H3 and 2s. And then finally, in 2009, it came a new pandemic virus. We call it pandemic H1N1. It's an H1, the same one as was circulating before, since 1918. But the H1 was very different antigenically to the H1 of the circulating. So this is what makes the virus be able to cause a pandemic that displaced the all H1N1s. And today we have circulating humans, H3 and 2s, H1N1s, and 2 lineages of influenza B viruses. So a total of 4 antigenically different influenza viruses. And because of that, the vaccines that we have, that they need to be used every year to update the components of the vaccine to match the circulating viruses, they have 4 components: 2 influenza B viruses, an H1 component, and an H3 component. Now, but there are more subtypes of influenza viruses circulating in nature. And from time to time, some of these ones, like H5N1s, H9N2s, H7N7s, H7N9s, they infect humans. These are infections that come in not from another human, they come from an animal reservoir. And these infections sometimes cause some severe disease, but it's only in a few individuals. And the reason is because there is no sustained transmission from these viruses from human to human. But if any of these viruses somehow acquire the properties of transmit well from human to human, then any of these viruses will cause a pandemic. Now, where these viruses come from, influenza A viruses, they are not only found in humans, but they are also found in many different animals. They are found in pigs, they are found in horses, they are found in dogs, in poultry, and in wild birds. In wild birds, there is a total of 16 different subtypes of influenza viruses, 16 different hemagglutinids. And these viruses are circulating in birds, wild birds. They're well adapted to these wild birds, but from time to time, they're able to jump into different species. For example, poultry. And if they start to transmit, they establish lineages in poultry. The same thing happens with swine, the same happens with humans, with horses, and with dogs. So most of the strains that are circulating in these animals, they don't jump from one animal to another. But when one of these strains jump and is able to acquire the ability to transmit, is when it's going to cause in humans a pandemic. Now, I'm going to start first about how we use new technology to study viral determinants of disease severity. And for that, I'm going to give you an example of a virus with which we have been working, which is the H1N1 of 1918. So this is the virus that caused the pandemic of 1918, a pandemic that was devastating and the worst pandemic that we know of influenza viruses in humans. Now, this study has been a collaboration between multiple groups, my group, together with a group of Chris Basler, Peter Palese here in Mount Sinai, but also in collaboration with the CDC, especially with Terry Tampi, the USDA, David Swain, and at the Armed Forces Institute of Pathology, where it was at this time, with our collaborator, Jeff Tabenberger. Now, what makes the 1918 pandemic influenza virus very unique? Well, I have 2 slides that show the devastating consequences of this pandemic. The first one is the US life expectancy in the last century by age, in which we can see that from 1900 to the 2000s, it has been increased steadily. And that's because of the modern medicine techniques, the use of hygiene, antibiotics, better hospitals, etc. But there was an exception, and this exception is this big dip that we have of more than 10 years, and that was caused by influenza virus. That was caused by the pandemic of 1918. The other slide that I have that illustrates the potential devastating consequences that this pandemic have, is this one that comes from a picture that is being in the Smithsonian. This is a picture of a village in Alaska that was taken after the pandemic 1918 influenza viruses has affected the village. And what is remarkable about this picture is that they have these people that are here in the picture, they are only kids. There is no adults. And that's because this virus, the 1918 influenza virus, has the ability, a bad ability obviously, to kill mainly adults that were infected with the virus. This is not because the kids were not infected, the kids were also infected, but in general they survived the infection, and the adults, especially in remote regions like Alaska, in which probably there was even less pre-existing immunity against any influenza virus, the consequences of the infection with this virus was very dramatic, and most of the adults succumbed from this viral infection. In other regions, not all the adults die, but there was also a high proportion of people that they were in age, healthy adults, individuals, that died of influenza virus infection in 1918. Now, why this virus was so special? Why this is not happening with other viruses that are circulating right now? Well, this one, we didn't know anything about what could be the reasons for that. And the main reason is because of 1918, it was not known that influenza was caused by influenza virus. There was no way how to isolate influenza virus at this time. And therefore, the first time that influenza virus was isolated was in the 1930s. And this virus, although it was a descendant of the 1918, obviously didn't cause some as severe disease as the parental 1918 virus. Now, the mystery of this virus could have been lost forever, is that there were some samples that were preserved where, although these viruses were not there as live viruses, there were pieces of the genome of the virus. And our collaborator, Jeff Taubenberger, was able to get the sequence of the virus coming from the samples. One set of samples came from lungs of soldiers that died in 1918 of influenza virus, and they were taking pieces, preserved in paraffin, and then stored in archives that they were maintained by the army, in order to allow future generations for looking to what could be the causative agent of a particular disease in soldiers. And I saw a picture of one of these paraffin embedded blocks of a piece of lung tissue from a soldier that died in 1918 of influenza virus. And the other samples came actually from a village similar to the one that I saw a picture, which is a village in Alaska, in which after the pandemic happens, after the virus came, then most of the adults died, as I saw also in the picture, and then these adults were buried in a common burial place under the permafrost line. So the bodies were frozen since 1918. They were not in very good state. The virus was not there, but then Dr. Helting came to this grave, dig the grave, and pick up some samples in which the virus was not there, but the genome of the virus was there. And with the genome, our collaborator, Jeff Taubenberger, was able to sequence the genome of this virus. Now, where new technology comes is that we have developed techniques that allow reconstruction of an influenza virus coming only from sequence. So this is based, we know the sequence. We can synthesize the genome of the virus using synthesis techniques for reconstructing the genome as DNA, and using what we call reverse genetic techniques that allows to go from DNA into RNA and come out with an influenza virus. Doing these reverse genetics techniques, we were able to generate back the 1918 virus from the sequence of the individuals that died in 1918 of Spanish influenza or 1918 influenza. And this was obviously that was done in high biocontainment units by our collaborators at the CDC. And now, having this virus reconstructed, we can characterize what are the unique characteristics of this virus, both in tissue culture and in animal models. So the virus was reconstructed, and this is a picture of the virus at the electron micrograph. And by the picture, you cannot see that this virus could be inducing a very severe infection. Neither the sequence, so any sequence that we know of at this time, that was responsible for severe infection with influenza virus, but this virus is very highly pathogenic. When this virus is put into animal models, among them, for example, ferrets, which are believed to be a good animal model for influenza virus because as humans, they can be infected with influenza virus and they have a disease very similar to the human disease. When you put in ferrets, if you put in ferrets a normal seasonal human influenza virus, and then you look to the lung of the ferret, you see a little bit of inflammation, but the lung looks quite healthy. So the animals have a cold, but it's mild, it's not severe disease. However, you infect the ferret with the 1918 virus. Now the lungs of these animals are characterized by a lot of hemorrhage, a lot of infiltration of immune cells that makes the lung full with fluid and pneumonia, high level of pneumonia, and the animal is unable to breathe anymore and then die of 1918 influenza virus infection. Now, we have been working with this virus now since we have reconstructed, trying to understand what makes this virus so different from other influenza viruses, and the summary of these results is expressed here in this slide. The 1918 virus is the only known human influenza virus that is lethal to mice, ferrets, macaques, and embryonic eggs. And we know also it was lethal in humans. So it's a very special influenza virus. But the reasons for this high mortality is not a unique reason. There are multiple genes that contains determinants of severe disease, including 4 different genes, HA, NA, NS1, and PE1F2, that have different functions during the viral cycle. So you could consider this virus a perfect storm. It just acquired it had multiple determinants of virulence that make the virus very special. That makes a virus like that very difficult to evolve again because it requires multiple determinants of virulence in order to acquire this virulence in humans. Now, the good news is that if we test existing antivirals against influenza, we can actually treat the 1918 infection in animals, as well as we use a vaccine based on H1N1. The vaccine is protective, meaning that the virus like 1918 we will be able to still be able to prevent infections with vaccines and to treatment with the existing antivirals. Now I'm going to switch now and I'm going to talk a little bit about the host determinants of disease severity. I told you about this virus, 1918, that is especially virulent, but seasonal influenza virus normally induce a very bad cold, but there is some people also that have a disease that is very severe and very similar to 1918. There are not so many people as in 1918, but there are some people that die every year of influenza virus infection. So what makes a person more susceptible or less susceptible to influenza virus infections? And we think that there are determinants in the host, in the human, that are responsible also for severity of disease. So there are non-determinants, for example, comorbidities, people that have cardiovascular disease, obesity, diabetes, immunodeficiency, a chronic respiratory disease. These are more likely to develop a severe influenza virus infection. And these are the people that they really need to be vaccinated in addition to the rest of the people, but these are the ones that get more benefits from the vaccine. Now, co-infections can also increase the ability of the virus to cause disease, especially secondary bacterial pneumonia. And other is the age, the old people, usually they have a more severe disease. Pregnancy also, pregnant women, they have a more severe disease in general. However, still many apparent young healthy adults that they don't get into these different groups die of influenza each year. Why? So we think that they have a predisposition in the genes that make it more vulnerable to influenza virus infection. And we're trying to figure out what are the genes that make you more vulnerable for some people than for some others in order to identify the people that may be most likely having severe influenza virus infection. So why is that? This is because influenza virus, like any virus, they need to use the cell to propagate. So the virus enters the cell and delivers the genome, RNAs, inside the cell. And inside the cell, it replicates and produces new virions that come out of the cell. And during this process, the virus is going to need to use some of the host genes in order to help in the propagation of the virus and in the multiplication of the virus. So by itself, the virus will not be able to propagate, requires host factors inside the cell, but also it needs to deal with antiviral factors because the cell responds to viral infection by producing antiviral factors that try to prevent the virus from replicating and killing the cell. And this is in this combination of how the virus deals with the host factors and with the antiviral factors, what at the end is going to tell whether one is going to have a more severe disease or less severe disease according to what is the sequence of these factors. Now, what are these factors? In order to identify these factors, we are using a systems biology approach. We call that our program Fluomics. And what we are doing is we try to look at the global level, and these are the people that are involved, the emails of the people that are involved in this program. We're trying to understand at the global level what are the genes that are used by the virus in using proteomics technique and genomics technique and metabolomics technique, and figure out what genes are important for the viral replication, both antiviral genes and required genes. And this is an example of what we can do. So here we have, in yellow in these circles, we have the different proteins of influenza virus. And what we have on the other circles that are connected with lines, we have proteins that are known, host proteins that are known to interact with these factors that are being identified through the systems biology approach. And in addition, they are represented here also not only the proteins that, host proteins that interact with these viral factors, but host proteins that if we eliminate them from the cell, they contribute to viral replication. So this is a set of genes, host genes, that now we have collected through the systems biology approach that we think are around 100 genes that we think they are very important for influenza virus replication. So we are looking whether there are human polymorphisms in these genes that are associated with severe disease. But the other thing that we can do with this information is we can try to target some of these genes for new antivirals. And that will be inhibition of host factors. In this case, I present an example here, a collaboration that we have with Ivan Marazzi here, also in Mount Sinai, in which we identified a host protein, SEC61, that is required for the export, for the transport of the virus glycoproteins that are important for making the virus in the cell. And this was identified through a proteomics approach as interacting with the viral glycoproteins. And there is an inhibitor called CT8 that can be used against this particular protein. When we use this inhibitor, then we're able to inhibit replication not only of influenza virus, but also of HIV and also of dengue without affecting the host. So that could be a potential good inhibitor that will be able to fight against multiple viral infections, not only against influenza. The last thing that I'm going to cover is the issue about influenza vaccines. And we have an influenza vaccine that works to some extent, but we need better vaccines. One of the reasons we need better vaccine is because when a pandemic comes, there is no time for making a vaccine that is against this particular strain of influenza virus that is able to prevent the disease and the transmission of this virus. This, for example, what happened in 1957 with respect of how rapidly, from the origin of the H2N2 pandemic, how rapidly the virus transmitted. So it was first detected in Southeast Asia in February 1957. In May, it was all extended by a large area of the world, including Oceania and all Southeast Asia. And in August 1957, it was extended through the whole world. And that happens in 1957, when there was less mobility of people by, for example, planes. And also the world was not so populated like today. So a pandemic today transmits with a speed that is even faster than in 1957. And what this means? This means that by the time that a new pandemic virus comes and you try to make a new vaccine, then the new vaccine comes late. This is, for example, what happened in the 2009 pandemic in the United States. We see in red the peak of viral infections during the first wave in 2009 of the pandemic H1N1. And we see in blue, we see the production, the shipment of the vaccine against this H1N1 virus. We knew how to make a vaccine, but to have the vaccine ready to vaccinate the people, it was at the time in which the first wave already went down. So the vaccine didn't make any impact in the first wave of the pandemic H1N1. It was helpful for the next second wave, but not for the first wave. If this pandemic would have been as devastating as the 1918 pandemic, this would have been a disaster. Fortunately, that was not the case. 2009 was quite benign. The virus quite benign pandemic. Despite that, there was also a lot of young people that died in 2009 of the pandemic influenza virus. One of the reasons is because we didn't have a vaccine on time. Now, how the vaccines are being produced, the majority of the vaccines that are against influenza virus, they are produced by taking a master high growth strain, putting the HA and NA genes of a circulating strain, then propagating embryonic eggs. The master strain grows very well in eggs, and the circulating strain provides the HA and NA genes. You harvest now a lot of viruses produced in eggs, and then you inactivate the virus and you purify partially the hemagglutinin and neuraminidase, and this is what is being used as the inactivated influenza vaccines. They are based on the hemagglutinin of the circulating viruses, because the circulating viruses change, the hemagglutinin change, and then you need to update the vaccine almost every year, and you need to take it every year. And again, if a new pandemic virus has, you need to go through all this process again, and because of that, you don't have the time to fight against the virus during the first wave through a vaccine against the virus. So the question is, can we develop a better vaccine, a vaccine that will be able to prevent infection with any influenza virus? And this is a strategy that we are now trying to explore in collaboration with Peter Palese and Florian Kramer, two investigators also here in Mount Sinai. Now, the concept that we have is based on targeting areas in the virus that are completely conserved. And what we are targeting specifically is a region in the hemagglutinin, which is here in red, that is called the stem of the hemagglutinin. The stem of the hemagglutinin is very well conserved, but it does not generate too many antibodies. Most of the antibodies, they are generated against the head here in blue, which they are very good antibodies, but that is strain specific. The head changes from year to year, and therefore the antibodies that you develop it for 1 year, they don't work for the strain that comes the next year. Now, the antibodies that one gets by natural infection or by vaccination against the stem, which are more conserved, they are very few. So we try to come out with strategies that will be able to induce more levels of antibodies against the stem than when they induce the vaccines that they are right now being used, or whether they induce natural infection with influenza virus. And for that, we use an approach that we call it repeated vaccination with chimeric HA vaccines. And I'm going to show you how this approach induces protective antibodies against multiple subtypes of influenza virus. And that's in collaboration, again, with Peter Palese, Randy Albrecht, and Florian Kramer here at Mount Sinai. Now, the concept is based on the following. We make a chimeric influenza virus hemagglutinin. A chimeric one is one that contains the stem from a circulating influenza virus, in this case, H3, and a head, which is in purple, and a head here in green that comes from an exotic HA, an H4. Humans, they have never been exposed to H4. We continue with now exposure to a different hemagglutinin that has a head from H5 and a stem from the same H3, and then at the end with another HA that is different, but contains also the same different head, but same stem. And at the end, we challenge with a virus that does not have, is completely different subtype, but shares the conserved stem of the H3 hemagglutinin. So what happened when we do this vaccination in mice? The first vaccination induced mainly antibodies against the head, but a few antibodies against the stem. Now, when we repeat it, because the antibodies against the stem, they are now in memory B cells, we are boosting these cells and we are inducing more antibodies against the stem, while we are priming again against a new head that is different from the green head. And now when we come with the next chimeric hemagglutinin, we boost even more the antibodies against the stem, that is common, but again, we do only primary response against the head, that is very different. So when we come back with a challenge that is an A7 and 9 that has the same stem, then the mice are protected. And that's what happened here. We see here that animals that are not vaccinated, they die, while animals that are vaccinated either with a vaccine that is matching or with this chimeric HAs, they have been protected against lethal challenge with influenza virus. But they are protected not only against H7, they are protected against H10 with a reduction in titers in the lungs, against H3s, either swine H3s or human H3s. And in fact, they are protected against all group 2 hemagglutinins, which they are here included, H4, 14, 3, 15, 7, and 10. Now, there is another set of hemagglutinins, group 1, including the H1s, that they are not protected against with a group 2 vaccine, but we can protect it if we use the same approach with group 1 vaccines. So if we use now a strategy based on a chimeric 9-1 following by 6-1 followed by 5-1, we're able to protect against H1N1. And by doing this bivalent vaccines, a group 2 and a group 1, we should be able to prevent any influenza A virus infection. If we add that in influenza B, then we should be able to prevent any influenza infection. Now, the protection is antibody-mediated, so we get high levels of antibodies measured by ELISA against the stem by this chimeric hemagglutinin vaccination, and we just take the antibodies and put it back into new mice. The animals are protected also against challenge. Now, based on this data, we started a prime boost vaccination using a live-attenuated influenza virus vaccine that is being used in some countries, followed by inactivated vaccine also that is being widely used, but based on these chimeric hemagglutinins. And we first did an experiment in ferrets in which we use a 9-1 followed by 8-1 and followed by 5-1, either as live-attenuated vaccines or inactivated vaccines. So we have different groups, one that gives the live-attenuated vaccine followed by the inactivated, another group that has the inactivated followed by the inactivated, one that is a control group, and we compare it with the actual inactivated vaccine that is called TIV and naive ferrets. Now, again, as I mentioned, ferrets are a good model for influenza virus infection. Now, when we challenge now with a virus that is pandemic H1N1, that does not have any of the heads of the vaccine, the first thing that we see is that there is a good levels of antibodies induced against the stock by the chimeric vaccines, either in blue or in green, that are high levels of antibodies after the last boost. And when we challenge with pandemic H1N1, then we get a nice reduction by the chimeric vaccines, especially the one that is live attenuated, followed by inactivated, in trachea, in the nose, and in the bronchi. And that's better than what we get, for example, with inactivated influenza virus vaccine. All right, so encouraged by these things, we have performed now a human trial, phase 1 trial still, for looking to safety and immunogenicity of this type of vaccines that are being funded, has been funded by the Bill Gates Foundation, in which we use a very similar approach to the one in ferrets. We compare a live attenuated followed by inactivated, that would be group 1. Group 2 is also live attenuated plus inactivated, but in the case of group 1 is with an adjuvant, ASO3, and in the case of group 2 is without adjuvant, the inactivated vaccine. So the adjuvants are supposed to give better immunogenicity to the vaccine. We have a placebo group, and then we have a group 4 that has inactivated, followed with inactivated chimeric vaccines, and a group 5 that is placebo. The live attenuated vaccines we put in isolation units to prevent potential spreading of the live attenuated virus, which at the end we found that that's not the case. This virus does not propagate between different vaccinated or not vaccinated individuals. And now, so the trial design overview is here. It was a prospective randomized control phase 1 trial, healthy male and female adults, 18 to 39 years of age, 69 subjects, 39 was done in Duke, and 26 in Cincinnati Hospital, randomized to one of 5 groups, median age 29 years, 40 female, 25 female. We didn't see any adverse event, and what we were able to now is to look for levels of antibodies against the stock. So in placebo individuals, we find some basal levels of stock antibodies that are, we don't think that enough for protection. Now, we use an approach that is based on live attenuated followed by inactivated. We see that after live attenuated, there is not so much thing happening, but after one gives the boost with the inactivated, one starts to see antibodies against the stock of the chain. If one use an inactivated adjuvanted, one get even a better boost. And the best results we have it when we use inactivated adjuvanted followed by inactivated adjuvanted. We get a very good response with the first vaccine against the stock of the HA that comes back a little bit, comes down, but then gets boosted again by the second. And these levels of antibodies, we know that they are protective for disease in animal models. But this is not only against H1s. We see that these antibodies cross-react with H2s, with H9s, with H18, with basically every group 1 hemagglutinin. So we are very encouraged by this data. showing that there is the potential that such type of vaccines will be able to provide protection in humans against any different subtype of influenza virus. So in conclusion, I show you that sequential vaccination with chimeric hemagglutinin vaccines induce a chain of stem antibodies and high levels of protection against heterotypic challenge in animals, and that this can be also induced in humans in this phase 1 clinical trials. Just want to show a picture of my lab. We call ourselves the flu fighters or the virus fighters, and some of the agencies that can contribute to financing the work that I presented here. BARDA, GSK, we have been collaborating with GSK, NIH, that is funded a Center of Excellence on Influenza Research and Surveillance that we call CEARS, as well as PATH and the Bill Melinda Gates Foundation that have been funding the clinical trials that I saw. So thanks a lot.